Welcome to this live webinar from AlphaLyse. The webinar is designed for CMC development and analytical methods for HCP analysis. So we are very happy to see the large number of participants also at this webinar. So welcome to you all. Welcome to our guest speaker, Victor.
Thank you.
So Victor is one of our most experienced bioinformatics scientists here at AlphaLyse. So the topic of today is experimental demonstration of the methods in the new USP chapter on HCP analysis. So Victor will go through the results from this study, and afterwards I will talk a little bit about the future perspectives. If you have questions during the webinar, please type them in the chat. And then Victor and I will answer them as best as possible afterwards. The presentation will take about 30 minutes, followed by approximately 15 minutes of Q&A. So just a few words on why we are here today and why AlphaLyse have come to this place. So we have been developing analytics for HCP analysis using mass spectrometry for many years. We had the first scientific publication in 2008 demonstrating how mass spectrometry could be used for HCP identification. In 2014, we started method development on how we could do identification and reproducible quantification using LC-MS. And in 2013, we started providing this as a service to biopharmaceutical clients. Since then, we have run a lot of projects on different types of biologics. We have gathered a lot of experience from different drugs and HCPs, and we have kept optimizing the methods. So these developments we have published in different ways and used in many different projects. In 2022, we got a GMP accreditation from the Danish Medicines Agency to perform validation and GMP release testing under GMP. And the other thing that happened was here in 2025, the USP published the final chapter 1132.1, where one of our main methods that we have been working on for many years is one of the methods in the chapter. So the USP also invited us approximately 1.5 years ago to demonstrate these methods experimentally, and Victor has been responsible for this study. So Victor will now share the results from this. So please, Victor, the stage is yours.
Thank you, Ivan. So my name is Victor. I'm a bioinformatics scientist and industrial PhD student at AlphaLyse, and over the last 1.5 years, we have been collaborating with USP to demonstrate the methods outlined in chapter 1132.1, and also to understand these methods and find their ideal use cases, but also understand their suitability for host cell protein analysis. So that's what I'll be covering in the next half an hour. So what's driving this change? Well, primarily there's been quite a few regulatory updates, and these are increasingly highlighting LC-MS for host cell protein analysis. And some of the key ones I've highlighted here on the left, with key guidelines from USP, but also from ICH. And overall, there's every indication pointing towards that both the industry and regulatory agencies are moving towards the use of LC-MS for monitoring host cell proteins. So why was this 1132.1 chapter even needed? Well, first of all, why is mass spec useful for host cell protein analysis? It enables simultaneous ID and quantitation, and this also enables knowledge-based risk assessment of particular host cell proteins, and it also enables comparison of results over projects. So this is what primarily is driving this with these benefits of LC-MS analysis. But LC-MS has been a little bit like the Wild West because of the vastly different quantitation methods, but also the very different instrumentation approaches which can be used. So there's been some need for some standardization to make this analysis robust and comparable. And that's more or less what USP 1132.1 set out to do. So why is this even relevant? Regulatory expectations are changing fast. So if you don't keep up, you risk being overtaken by regulatory requirements for LC-MS HCP analysis. So it's really important to consider this. So 1132.1, it's something I've mentioned 3 or 4 times now, and I would briefly like to cover the most important aspects of this, since regulatory authorities are going to refer to this chapter, and therefore it's quite important for you to know and understand it as a CMC mentor. So first of all, as in most LC-MS-based analyses, there's a sample preparation step. Here it covers quite different components, but overall it requires a proteolytic digestion to break proteins into peptides so they can be analyzed by LC-MS. There's a reduction and alkylation step, and then addition of some kind of standards. This is followed by a chromatographic separation so that these peptides can be separated and analyzed by the mass spectrometer. Then there's the mass spec part. There's no specific instrument or acquisition method which is mentioned as the optimal way. Overall, 1132.1 says that you should consider all the different options available and choose the one which is best suited based on what outcome you would require. So think about what data you would need at the end. And there's quite a few different instruments to choose from, which can be considered. Then there's the data analysis and quantification aspect. There's quite a lot of different analysis software and algorithms available. USP 1132.1 covers no specific software, but says again that you should consider what you want out from this and use the one which makes sense for you. But when it comes to the quantitation algorithms, or relative to, there are 3 options mentioned here. There's method A relative to product protein, B relative to spiked in protein, and C relative to spiked in peptide. So this is what I'll be diving a little bit more into in this study. And in this study, USP approached us to collaborate and practically demonstrate these 3 methods so that we better could understand them, but also find out what are the advantages and disadvantages of each of them. And overall, like I mentioned earlier, we have the relative to product protein method, where you use the product protein peptides for whole cell protein estimation. Then there's the relative to spiked in protein method, which uses known protein standards spiked in known amounts. And then there's the relative to spiked in peptides, where you use stable isotope labeled peptides and also spike them in known amounts. So how did we use this in the study? Overall, good monitoring of high-risk whole cell proteins will help you avoid clinical trial setbacks and regulatory delays. So it's crucial to have a method capable of identifying and monitoring these whole cell proteins and others like them. To demonstrate this, we selected 2 whole cell proteins, live protein lipase and clustrin, which are known to be high-risk impurities, and LPL, which is known to degrade polysorbates, affecting biological drug stability, and clustrin, which is immunogenic and can have some severe immunogenic reactions. So these were quite important whole cell proteins to monitor. So we used them as a to move forward. To do this, for the study itself, we combined a USP map 0.03 with a null HCF, which was known to contain LPL and clustrin. Here, peptides from the protein, or the map, was used for this method. This was followed by spike-in of intact protein standards, to be exact 7 different ones, which would be used for method B. In this case, we would use beta-lactoglobulin as the reference standard for quantitation. This was followed by reduction alkylation and enzymatic digestion, whereafter SIL peptides were spiked in. And in this case, 3 different cell peptides for each of the target host cell proteins were spiked in. This was followed by LC-MS analysis on 2 different mass spectrometers within acquisition methods, a untargeted approach versus a targeted approach. The data was then set through some identification and integration, and there was then a quantification and evaluation aspect of it as well. Here we evaluated the data against ICHQ2-R2 validation parameters, looking at accuracy, linearity, precision, and specificity, so we could get a good comparison of these 3 methods from the same data set. And then we will also assess the method advantages and limitations based on the real world observations, I would say. So, as mentioned, the data was analyzed with these ICHQ2 guidelines in mind. And it's quite important that these are fulfilled so that you don't go through the trouble of working with a method which in reality can't be used for regulatory documentation. So it's quite important that these parameters could be used. So to start with, we can dig into linearity. Here we have the concentration on the x-axis and the MS area on the y-axis, and we see for method A relative to product protein that it has an acceptable R-squared value or linearity across at least 5 points. There is a little bit of trouble at the start, but overall it still passes the criteria we set up. Relative to spiked in protein, method B achieved excellent linearity across quite a wide dynamic range. And relative to spiked in peptide, also achieved excellent linearity. So overall, the rule is like no linearity, no useful quantitation. So linearity is essential for ensuring comparable and useful quantitative results. The range is also quite critical to consider. So method B, for example, obtained linearity across 4 orders of magnitude, which means that you essentially can trust this quantitative data both at the very low but also at the very high levels of host cell proteins. Moving on, then we assessed precision. Here the data is from intermediate precision, which is shown across 9 different measurements from 3 different days on 2 different instruments. We overall see quite different quantitative results depending on the method, but also the standard used. As you can see here, depending on the peptide set used for method A, we get different results. And also depending on the cell peptide used, we get different results. Same thing is reflected for both target host cell proteins. However, the important thing here is that the CVs, or the coefficient of variation, are very low. So this means that when you repeat the analysis, you will get the same result, and generally below 20% is considered very, very good. And as you can see here, they're very, very low. So overall, this very high reproducibility or high precision means that the results are reproducible. The results won't vary due to instrument or analysis variability, and you'll be able to compare your results to show your process is consistent or use it for process optimization. But it also means that dependent on the reference standard or method used, you will get different results. So it's important to stick with the same one. So the results as shown here are from the untargeted approach. The targeted approach also achieved complementary results. We then assessed specificity as well. So the importance of this is to control that the spike in standard does not affect the HCP quantitative results. And to do this, we did a comparison between a spiked and an unspiked sample to see if there was any interference. And overall, it was quite clear that, again, looking at the excellent CVs, that there was no interference observed. Yet a single peptide stood out, both when looking at t-test, but also at the CV, indicating that there was some interference with the quantitation on this. But overall, it's essential to ensure that results are not affected by reference standards used. So for that specific reason, we run client samples just to be safe, with and without a spiked-in standard, to ensure that there's no interference from our standards. And again, the targeted approach achieved complementary results. So what should you consider when choosing which method to use for your biologics project? Relative to product protein method A, it has some quite specific advantages. Overall, it's applicable to all host cell proteins in the sample, so it's not bound to any specific host cell protein, which makes it quite desirable. It normalizes for sample preparation variability. The drug product is present throughout the entire sample preparation process, so it will be under the same conditions as your host cell proteins. So that accounts for some of the sample preparation variability. The sample setup is quite fast, making it easy for fast exploratory analysis. However, it is only suitable for early discovery, which would be one of the downsides of this method as well. On the limitation side, there's a large dynamic range between the product protein, but also the HCPs. So that can cause some issues. It's difficult to compare across different sample types and product proteins because, as you saw in the earlier figures with the precision, for example, depending on the reference standard and the method used, you will get different results. So if you want to look across some different projects or different biologics, this would not be possible with this method. Unexpected modifications in the product protein can affect results. So if the peptides you're using somehow get affected or have a modification, this will and can affect your results. For method B, relative to spiked-in protein, it is also applicable to all host cell proteins in the sample. It also normalizes for sample preparation setup as it is spiked in prior to digestion. The sample setup is quite simple, as it's with known spike-ins. This method is suitable for process and product characterization. It also supports the transition to a target MRM approach, which is one way it discriminates itself from or stands out from method A, which, due to the high dynamic range of the product protein, cannot be used for a targeted approach. It enables a universal method where you could have the potential for a generic reference protein because you would be able to use it across different projects as it is spiked in. There are some limitations, however, as well. It also requires a spike-in process, but also comparison with and without spike-in to ensure there's no inference. And this is something which cannot be done for method A. The untargeted method has a slightly higher lower limit of quantitation compared to the targeted method when using relative to spiked-in protein method. Okay. Method C, relative to spiked-in peptide. This is only suitable for targeted approaches. It is effective for specific host cell proteins of interest. So if you know you have some high HCPs of concern, monitoring them with this is an effective way. For example, clustering or light LPL. Pre-made isotopically labeled peptides are available for some known host cell proteins of concern, again, like clustering and LPL. But there are some limitations with this method as well. For example, it relies on complete proteolytic digestion of the peptide, recovery of host cell protein. So because it assumes that there's a complete proteolytic digestion of the host cell proteins, the results may not always be comparable because it depends on how effective your digestion has occurred. Spike-in timing pre or post digestion will also cause variable peptide losses. Synthesis of peptide stability may also vary. So depending on the peptide you have and the synthesis stability of this, your results will also vary over time. So overall, what are the conclusions from this? So which method is best? And this, of course, depends on the purpose, but in our opinion, and supported by the results from our study, one method definitely stood out. So just to summarize, LC-MS identifies and quantifies each individual host cell protein. 2 out of the 3 USP-NF methods met the validation parameters from ICHQ2-R2 we set up for this study. And method B, using spiked-in protein standards, is, in our opinion, the most ideal method. And this is supported by the experimental evidence of our study that the analysis relative to spiked-in protein is the preferable method out of these methods outlined in 1132.1, as this method can be used throughout the entire product development process, from early discovery to release testing, but also can be used for targeted approaches. But this also means that it can be used as a platform approach, as it is not bound to a specific product protein, organism, or requires synthesis of peptide standards. So overall, this means that this data can be used for regulatory submission against the same reference standard, and is why we believe it to be the gold method for quantifying host cell proteins. Now, this was a brief overview of a lot of the work that myself and the people on the listed here have been doing. So if you would like to dig into a lot more of the work, you're more than welcome to have a look at our preprint, which hopefully will be accepted as a fully peer-reviewed article within the near future. The paper itself, it was more or less a practical guide to implementing USP 1132.1 in your workflows, and goes into some of the deeper details of how to do this. So with that, I would like to hand over to Ivan, where he will be digging into what this actually means for the future.
Mm-hmm. Thank you very much, Victor. Quite a substantial work and quite impressive. So I can say that as one of your PhD supervisors. Yes, please have a glass of water. So what are the future perspectives of this new USP chapter and the methods we have just seen demonstrated by Victor? What will it mean for your projects, and will it change regulatory requests for LC-MS data? We think it will. So each year we go to a conference called the BEPA Host Cell Protein Conference. So we went again this year in May. And what we are also always very interested about, and what we always asked about, what will regulatory authorities say about the use of mass spectrometry for HCP analytics? So normally the presentations from EMA are on ELISA radiant characterization, and mass spectrometry is not really mentioned. But this year, Erika Friedl from Paul-Ehrlich Institute in Germany, which is EMA in Germany, presented several slides containing mass spec expectations. So Erika said, now there is an increasing regulatory focus on individual HCP characterization and control. So individual HCP characterization means you have to use mass spectrometry to identify the HCP. Otherwise, you can't know the individuals. She highlighted the use of LC-MS for identifying and quantifying HCPs, doing process development, process changes, and also manufacturing site transfers. Monitoring of individual high abundant HCPs in the final product is also a focus. As a biologics developer, you should always use a knowledge-based approach for problematic HCPs, and she referenced several recent publications on problematic HCPs known to degrade your product or known to be immunogenic in patients. So you are expected to use the current new literature. Also, she referred to the new USP chapter 1132.1, describing the mass spec methods, and advocated you should use the best practice methods. She also got a question from one in the audience. Now there are all these methods, which ones should we choose? And the answer for that is that you, as the biologic developer, you have to present your product and your process to us and show that you have a good control strategy on HCPs. You are expected to use best practices. You will have time to catch up with these new methods. You will have time to catch up. It means in the future you will be expected to provide these. So although it's not a formal requirement yet in the guidelines to use mass spectrometry, we see the effect already on several client projects when they come to us. And here I've just given a few examples. One of them is from China on a market submission on a monoclonal antibody. Here the client was asked to document if there were known problematic HCP in their map and to demonstrate that the ELISA was capable of detecting these HCPs. So we did a characterization of the ELISA reagent using our method ELISA-MS, which you can also find more information about at our website. The second example is from FDA. It's a recombinant protein already on the market. So there is one HCP that is known to be problematic. Here FDA required that there was a specific method validated and capable of doing GMP analysis on this specific problematic HCP. So it was a requirement to have this product on the market. The third example is a vaccine, a request from EMA. It's a vaccine that is already on the market. The current ELISA is not very good. The coverage is quite low. So here EMA requested that an orthogonal method was used, namely LCMS, on all GMP batches to confirm the HCP level in all batches. So although it's not a formal requirement in the guideline, when you meet these questions from regulators, then you have to do mass spec to answer the question. So we think we will see more and more of this in the future. So what method should you choose? Of course, as Victor just demonstrated, we believe here at AlphaLyse that we have the best, most global methods for doing this. So when we do relative to spiked in protein, this method works for any HCP quantified against this reference protein, and it works for any type of drug substance. The other two methods, method A relative to the drug substance, it only works locally for this drug substance. Method C, it only works for pre-selected HCPs of concern. So of these three methods, the one in the middle is the most global one. We have already used this method since 2017. We have set up a platform to do this. We have automated a lot of the analysis, and we have run now more than 650 different projects. So these are spread over different types of modalities: recombinant proteins, monoclonal antibodies, vaccines, cell and gene therapies, and even products, non-protein products containing proteins. We have analyzed more than 50 different organisms. The method works for all of them. In total, we have analyzed thousands of samples, and we have seen more than 1 million of HCPs molecules. So a lot of experience, and with this data collected on a platform method, we are able to compare results over time when you send new samples or from project to project, between projects, and even between other similar drug types from other developers. Are the HCP levels you observe similar to other products? So what we see in general, how our clients apply these techniques, they apply it throughout their different drug development stages. In the preclinical, you do it to quantify the total HCP level, to see if there are abundant or problematic HCPs that were not detected by the ELISA that may be of a risk to patients. At the BEPA Wholesale Protein Conference, Novartis actually stated, when first time you go into man, you're always a little bit concerned. Are there safety risks? Could there be some abundant or problematic HCPs that your ELISA didn't detect? So just to be absolutely sure, that they decided that they will do mass spec on all their preclinical samples before they move into man first time. When you go into the clinical development, our clients use the technique to do process development to control and remove abundant or problematic HCPs, and they use it for batch comparability when they change the process and when they scale up. In phase 3, the method is used for process consistency for PPQ runs for market licensing applications. The method can also be validated, and we can do GMP release testing on clinical batches throughout the clinical phases, and we can also do it for QC routine testing for products on the market. So this strategy many of our clients have used and are using. So we have actually qualified and validated a large number of projects and are doing QC routine testing here at Alpha now. So you can see it's a variety of different types of modalities. It's vaccines, recombinant proteins, cell and gene therapies, MAPs, enzymes used as part of the manufacturing process. All these samples, different sample types can be handled by this method. So when you look at this in overall, what are the future perspectives of this new USP chapter? The chapter and the methods in the chapter, it enables direct comparison of ATP levels across samples, across projects, and even between laboratories and between different drugs on the market and under development. So now we can compare results from project to project. It also enables risk assessment of the individual HCP across the industry. So in the future, we will know what level of HCPs are safe to have, what have patients seen before, are these levels problematic or not, and we can compare between different laboratories because we use the same method and the same reference standard. It also enables a release testing using LC-MS only. If you have a good MS method and you can apply it, there's no need to go and develop a process-specific ELISA. It typically takes 2 years to develop a process-specific ELISA. The mass spec platform is ready to use. And finally, the method is ICH compliant. It's a platform method. When we do validation, we do a process-specific validation on your product and your impurities, but it is a platform method that is validated. So we believe here at Alpha that this is going to change the future strategy for projects. So that was my last slide. So I'll invite Victor in here again, and then please type in your questions in the chat, and Victor and I will try to answer them. So I'm just reading here the questions. Victor, can you click on the Q&A? So some different questions here. So for linearity, Victor, did you force y-intercept to 0? And yes, why did you do that?
Well, I actually did both. I did with and without to see what impact it would have, and I ended up using a forced y-intercept at 0 because it didn't have a big impact on the calibration curve in the end, and because it should end at 0 at some point. So yeah, there's no other real reason than that.
Then I have another question here that I think is quite typical. I've read the new USP chapter, and I didn't really understand it. So how will your manuscript and article be useful, Victor?
So hopefully, the idea with the manuscript itself was, of course, to compare the three quantitative methods, but it's also to give some clarification into the practical application and how you could implement this into your workflow. So it thoroughly describes each of the three methods, the quantitative algorithms behind it, enabling some better understanding of how they work and can be applied, but also different parameters which can be essentially it enables you to assess which of the three methods would be suitable for you. That's the overall purpose of it, and it gives you a more practical perspective on them compared to USP-Learn 32.1, which is a bit more general and doesn't necessarily describe any specific instrument or any specific algorithms surrounding these quantitative methods. So it clarifies a lot of these things surrounding that.
Thank you. Another question here. In your linearity plots, though the data appears linear, what was the recovery and percentage CV at low concentrations? The plot you showed may have been biased by the higher concentration, making the plot appear more linear. Is there a different way to plot these data?
Yeah, so you can also log transform both the y and x-axis to assess linearity, so that the separation of the points don't bias this. Because of course you can have all the lower points in the lower end, and then a point very far away at a higher range, which could make it look linear without it actually being linear. But by log transforming it and assessing this, then you can see if it actually is linear. So of course we also did this to ensure the linearity of it. But when you do log transform it, then you can't use the calibration curve for quantitation. So yes.
You mentioned you run all samples both with and without spike in to look for interference. Would it be possible to determine, do this in silico instead of running the unspiked sample?
Theoretically, maybe. I'm not sure, to be honest. Of course, you have to see. You could try to do it theoretically or in silico, but I would personally always want to see it myself, because how does the mass spec actually react to this and the quantitative algorithms processing the data? So, in my opinion, it is necessary to do it.
You're a bioinformatics scientist, yes, and you have a huge data set.
Yes.
Where you did both.
Yeah.
Could you I don't know, answer this question by looking at all the data you have collected?
It's a good question.
So we'll dig more into this in the future. Do you have any suggestions for selecting a suitable protein spike, and what protein spike in standard did you use at AlphaLyse?
So, can I get the question again?
Yeah, how do you select a good protein spike in?
Spike in level or spike in standard?
No, spike in standard.
Okay, so there's a few different criteria which are necessary. First of all, there of course should be no interference with the target host cell proteins. So if you're using a CHO cell line and you use CHO spike-in proteins, that would probably cause some issues. So I wouldn't do that. The other thing is the protein has to be within the linear range of the instrument. That is another very important fact, because if it's not linear, then it's not quantitative either. So that's another important aspect. Those are some of the key considerations, at least. There are some other ones as well, but that would make it a lot more specific. But to answer your question with regards to which standard we use at AlphaLyse, we use beta-lactoglobulin as the main reference standard for all our quantitation in the samples. And that's what, as you saw in the precision slide, when you use the same method but also the same reference standard, you will get very, very comparable results. But as soon as you change the standard. And even with cell peptides, if you use 3 different cell peptides from the same target host cell protein, you will get different results. So it's very important using the same one. And that's why we've been using beta-lactoglobulin for all these projects. We of course also have other proteins in there to assess performance and so on, but beta-lactoglobulin is the primary one.
Mm-hmm. And the next question is also a great one. So could you comment on post-translational modifications on the HCPs when using protein standards, effect on digestion efficiency and quantification results if the spiked protein has different PTM profile than the actual HCPs?
So that is something which definitely would have an impact. So let's say you're using spiked in cell standards, which in that case would be unmodified, and then you're trying to quantify your target host cell protein where the peptides you are targeting are modified. This will definitely skew the results. However, you could look for the modified version, but using that specific approach, it would cause some issues with regards to general host cell protein quantification. At least for our setup here, we did not include modified peptides other than oxidation of methionine and carbamethylation, but because that would vastly complicate, make things much more complex. So, but it is definitely something you should consider. Yeah.
Mm-hmm. Yeah, thank you. How is your experience with very low abundant HCPs? Do you have special workflows for that, for example, for maps or?
So generally for very low abundant host cell proteins, a targeted approach would be ideal because you can select specific peptides which have a better ionization efficiency. So they will be detectable in very low amounts. And by selecting those for a targeted approach, you will be able to monitor them at very low levels. And that's also something we demonstrated in the manuscript, where we where the method was linear all the way down to 0.2 nanograms per milliliter for the targeted approach.
Mm-hmm. And then I think we have the last question here. Have you compared ELISA results with LC-MS results? Should we expect the same amounts measured?
That's a very good question. First of all, I'm almost 100% certain you will not get the same results. ELISA is based on a reference standard, so it's always relative to that. The same thing with mass spec. And as you saw on the slide comparing the precision of the method, depending on the reference standard used for mass spec, you will also get different results. So the results will be very might be different, but it could also in theory be comparable. But the important thing is when you use mass spec, you'll have the possibility to look across different projects using the same reference standard, where this will not be possible for ELISA. Mm. So with mass spec, you can compare and look across many different projects using the one and same method.
Mm. This exact question is actually also addressed in the new USP Chapter 1132.1. That when you use ELISA, when you use mass spec, you should not expect to get exactly the same quantity values. So they use different principles, they use different reference standards, so the amounts are not, they don't have to be exactly the same. So with that, Victor, I will let you off the hook, and thank you to all the audience. If you have any questions for us, don't hesitate to contact us afterwards. Then Victor and I will be happy to take your questions and answer them by email or a Teams meeting. So thank you very much for your participation and your great questions. Bye-bye.